Hey guys, Dan here. What we're going to be doing today is exercise 2, cellular anatomy and mitosis. Let's get into it. So what we're going to be doing for this exercise is object A found on your handout, and what we're going to be doing is going through each organelle and their function, as well as pointing them out on the model. Some of the models are not completely accurate in showing all organelles in here, so just a helpful hint, we're not going to be testing on anything not found on the model. First up, we're going to have the nucleus. Now everyone knows what that is, but let's just double check this. So the nucleus is everything found underneath the nuclear envelope, including the nuclear pores. What this does is the genetic control center of cellular activity, and that's found right here. Next, we're going to have the nuclear pores. As just stated, they're these little holes right here coming from the nucleus, and what they do is regulate molecular traffic in and out of the nuclear envelope. Secondly, we have the plasma membrane, and what this does is regulates traffic from in and out of the cell by facilitating diffusion, but the main function of the plasma membrane is protection, and what that does is just a barrier from outside forces. Ribosomes, found as these little red dots, and I'll try to get a little bit closer right here and then also on the rough ER, which we'll talk about later, and what those do is synthesize proteins. Next, what we're going to be having is the smooth endoplasmic reticulum, also known as the smooth ER, and that is the ones without the ribosomes found right here. What the smooth ER does is it synthesizes lipids as well as steroid hormones and detoxifies drugs. Additionally, we're going to have the rough endoplasmic reticulum, known as the rough ER, and that's found right here with the ribosomes. And you would guess it, as it has the ribosomes, it's going to make proteins as well as package and fold them. The Golgi apparatus is not found on this model, however it is an important organelle found within the cell. The function of said organelle is going to be secretion and intracellular transport. So next what we have is the lysosomes, not found on the model as well. However, another important organelle aids in the function of hydrolyzation of proteins, nucleic acids, complex carbohydrates, and other substrates. However, my favorite function of a lysosome is it aids in apoptosis, known as cell death. Peroxisomes, not found on the model, utilize O2 to oxidize organic molecules. So everyone's favorite, the mitochondria. Please do not say the powerhouse of the cell. That is not entirely correct. The reason why it is the powerhouse of the cell is because it synthesizes ATP, which is a lot better of an answer. And those are found right here. And you can see they come in all shapes and sizes. So the cytoskeleton is made up of microtubules and microfilaments, which we'll talk about in the next two sections. And what those do, it gives the cell shape as well as motility and directs the organelles where to go, additionally to keep them in place. So you guessed it, microtubules not found on the model. However, they use our transport from mitochondria and other organelles within the cell. They're utilized by moving them, transporting them around the cell, as well as giving the cytoskeleton a little bit more structure. Microfilaments, on the other hand, make sure that the organelles are kept in place and then also allow for cell movement. Additionally, we have cilia and flagella, and what those do are the whip-like little kind of things on the back of, say, sperm that allow for the little tiny particles to move, and they use it in kind of a whip fashion to move around the cellular material. Next, what we have is the centrioles, found on the model finally, and what they do is they aid in cell division found in mitosis, and these are these guys right here, and they look like little stars, or the kind of the candy that you ate as a little kid. So what we're going to be doing next is we're going to completely skip part B for obvious reasons, and then skip to part C, which tells you to make sketches of the following cells. We'll do those in class, so I won't have to provide any models. Next, what we're going to be talking about is mitosis and the process of cellular differentiation. The steps of mitosis are found in interphase, prophase, metaphase, anaphase, and telophase. The best way to remember them to make sure that you keep them in order is mnemonics such as, I passed my anatomy test. That's just a way to make sure that you keep those letters in successive order and making sure that we can learn from that as well. So interphase, found right there, the very first one, it was also known as the resting phase, and that's when the cell's kind of duplicating its DNA, getting ready for the meiotic division. In anaphase, the spindle fibers have completely taken apart the centrosome, pulling the chromosomes into two parts, allowing for the two cells to start becoming. This means that the chromosomes are completely broken, as well as the spindle fibers. So prophase is kind of when the centrioles are moved to the poles, the spindle fibers start to grow, and then also what we have is organelles breaking down. So finally in telophase, what we're going to have is that kind of cut between the two cells that have now appeared, thus giving you two new cells. So just as a final note, what you guys are responsible for is making sure that you look up all the structures and just being able to identify that. So even though I won't test on you, it's still a very good idea to make sure that you do know what you're doing in terms of looking at any model and being able to determine what they are and what their functions are. Additionally, what we're going to have you do on this is also look up what each phase of mitosis looks like itself. We're going to have slides for you to look at, but it's a good idea to come to lab knowing what you're doing before showing up, just so you have a better utilization of lab time. So that's the end of exercise 2. Please email me if you guys have any more questions. Thanks, and bye.